Hello everyone, my name is Maria Mendoza and I'm one of your TAs for the spring of 2022, and this is your exam to review. So we're just going to be going over the two exams that your professors have uploaded, one from Professor Dudagachi and the other one from Professor Zogby. We're going to go over Dr. Dudagachi's first. Now, anytime that I reference a video, because a lot of this material, I already made videos that explain those concepts in more detail. Those will be on my YouTube channel, and you can find them linked in the announcements. Okay, so here are some formulas that she gave you. pH is equal to pKa plus log of the acid over the base. And then delta G is equal to delta H minus T delta S. And then the Michaelis-Menten. Okay, so question 1. The figure below shows the Lineweaver-Burke plot of two catalyzed reactions of an enzyme with and without an inhibitor. Which of the following is true for the figure? So for a Lineweaver-Burke plot, it's really important that you know the axes. So for the x-axis, you have 1 over the substrate concentration. For the y-axis, you have 1 over v. And then wherever your line intercepts on the x-axis, that is 1 over km. And then wherever your line intercepts on the y-axis, that's your 1 over your Vmax. And then from my notes, I went ahead and from the lecture notes, I went ahead and copied and pasted the different types of inhibitors that you could have, along with some definitions. So there's really 3 types of inhibitors. We have competitive. Let me grab my pen. Okay, so we have competitive inhibitors, we have uncompetitive inhibitors, and we have mixed. Now this non-competitive inhibitor is actually a subtype of mixed inhibitor. Now for competitive inhibitors, okay, let's go ahead and read our definitions. So these are reversible inhibitors, and these are 3 types. So competitive, uncompetitive, and mixed. And you have to know their little graphs for this Lineweaver-Burke plots. So for competitive inhibition, the inhibitor binds to the active site. So the inhibitor, for competitive inhibition, the inhibitor binds the active site, and it prevents the substrate from binding. The apparent Km, and yeah, that's the apparent Km, and it is the Km that you see when the inhibitor is present. If you add more substrate, then you reduce the chance that the enzyme will bind to the inhibitor. The substrate concentration will have to increase to achieve the same Vmax. So you can see here that this black line is when there's no inhibitor, and then this red line is when you add an inhibitor. So when you add an inhibitor, the Vmax remains the same. So they both lines cross the axis in the same location. So the Vmax does not change, but the Km increases. You can see that it moved to the right with the inhibitor. And this right here is 0, so anything on the left of this 0 is negative. So the line became less negative, which means that the Km increased. For uncompetitive inhibitor, you can see that whether you have no inhibitor or whether you have an inhibitor, you just have parallel lines. So both the Vmax and the Km, they both decrease. And then here for non-competitive, which again is a type of mixed inhibitor, the x-axis, where the lines cross on the x-axis, remains the same, and that's the value for Km. So Km does not change, but Vmax decreases. And then when you have a normal mixed inhibitor, both the x and y intercepts change. The Km increases and the Vmax decreases. Okay, so they're asking which of the following is true for the figure. So if you look at this figure, we can see that whether you have the inhibitor or not, they both cross at the same y-intercept, but the x-intercept is different. And if we remember from our graphs, we can see that this is a competitive inhibitor. And so we can see that this dashed line is the inhibitor, and this is no inhibitor. Okay, so let's read our choices. The solid curve belongs to the enzyme with a competitive inhibitor. So no, that one belongs to the no inhibitor. Let's look at C. The dashed curve belongs to the enzyme with an uncompetitive inhibitor. No. So we decided that this is competitive. And then D, the solid curve belongs to the enzyme with an uncompetitive, and again, no, because we know that this is competitive. So B, the dashed curve belongs to the enzyme with a competitive inhibitor. Yeah, the dashed one. So that's how you figure that out. Okay, number 2. The figure below shows the Lineweaver-Burke plot of 2 catalyzed reactions of an enzyme with and without an inhibitor. The V0 for the inhibited reaction if substrate concentration, okay, so they're telling you V0, so you have an inhibited reaction and a non-inhibited reaction. And so then they give you values for these. So the substrate concentration is 12 micromolars. The y-intercept for A is 0.4. The x-intercept B is 0.25. And the x-intercept C is 0.45. So down here is the graph that they're talking about. So remember, whatever crosses at the y-intercept right here, that's the Vmax. And then whatever crosses at the x-axis is the Km, or actually the inverse of both of those, because this is a Lineweaver-Burke plot. And so then, let's see here. So again, we kind of have a clue, and we can see that both of these lines are crossing at the same y-intercept. So that means that we all automatically know what kind of inhibitor this is. So we just have to go up here and remember that this is a competitive inhibitor because the y-intercept is the same and the Km increases. So the fact that this is a competitive inhibitor means that they're going to have the same Vmax, but they're going to have different Km values. So we just have to find one Vmax with the numbers that they gave us, and then we'll be able to solve this equation. Okay, so I went ahead and wrote down the numbers that they gave us. So they gave us the y-intercept is equal to 0.04. Yeah, that was a y-intercept. And then the x-intercept was 0.25. And so all you have to do is take the inverse, because the y-intercept is equal to 1 over the Vmax, and you set that equal to the value that they give you. Okay, so yeah, the y-intercept is equal to 0.04, so 1 over Vmax is equal to 0.4. So then you just swap these, and that's 1 over 0.4 is equal to the Vmax, which is equal to 2.5. And then for the x-intercept, they gave us that it equals 0.25. So you just do the same thing. 1 over Km is equal to 0.25. And then you just swap these. So that's 1 over 0.25 is equal to Km, and that equals to 4. And so Now we can go ahead and insert our values in this formula. So this is the formula we're given. V naught, that's the initial velocity, is equal to V max, which is the maximum velocity, times the substrate concentration, which they gave us, divided by the Km, which we figured out mathematically, plus the substrate concentration again, which they gave us. So now we just plug in. The numbers that we were given. So it's 2.5 times 12 micromolar plus, and then 4 plus 12 micromolar, and then once that will give you 30 on top and 16 on the bottom. And when you divide, V naught is equal to 1.875 micromolar per second. So we can see that that answer is right here. So also, we have a video on YouTube for the Michaelis-Menten stuff. If you need help, you can go look at that. But I'm going to cover just some important terminologies. So what is the purpose of the Michaelis-Menten equation? It shows the relationship between the initial velocity, which is V naught, and the substrate concentration. Now, definitions to remember. Km is the substrate concentration at half of the maximum velocity. So for example, here we have the maximum velocity there, and then so half of the maximum velocity would be here. And so if you take that all the way down, that number right here will be your Km. And remember, the x-axis is the substrate concentration. So here they're giving it to you in millimolar, and on the y-axis we have the velocity in micromolar per minute. So anytime you have to do math, make sure that you convert. Okay, so we have our Vmax, half of Vmax, and our Km value. Now, the Vmax is the maximum rate of the reaction. At this point, the enzyme is saturated. So if you are, sorry, that's my dog. If you are at the Vmax up here, then the enzyme is saturated. So let's say that there's 100 enzymes total. That means that they are all full with substrate. That is what saturation means. And no matter how much substrate you add at that point, the reaction can't go any faster. So if you notice down here on the x-axis, the farther you move to the right, the more substrate that you are adding. But at some point, everything becomes saturated, the enzyme becomes saturated, and you will no longer increase in your velocity. So that's why it's kind of stabilized here. V naught is the initial velocity. This is dependent on the substrate concentration. So the initial velocity is dependent on substrate concentration. If you have a little bit of substrate, your V naught will be low. If you have a lot of substrate, your velocity will be higher. And then we have the catalytic efficiency number, the kcat, is equal to the Vmax divided by the total enzyme concentration. Now, ET, that's total enzyme concentration, that includes the amount of free enzyme plus the amount of enzyme that is in a substrate, that is in a complex with its substrate. Okay, so now we're going to cover 3 different situations. So the first one is this red dot right here at the very beginning. So you can see that here we have low substrate concentration, because that's all the way on the left on the x-axis. So we have low substrate concentration, and you only have a little bit of substrate. So on the x-axis, you can see that the Km, so this Km value over here on the right, is greater than the substrate concentration. It looks to be close to 0. So when you look at the Michaelis-Menten equation in the denominator, where you would normally add the substrate, you're adding almost 0, so it becomes negligible, and they remove it. So they're talking about the equation right. Okay, so they're talking about this equation here. So notice that this is the normal Michaelis-Menten equation. So on the bottom you have Km plus the substrate concentration, but here they remove the substrate concentration because there's almost no substrate. It's almost 0. So that's why that looks that way. Okay, kind of messed things up. Hang on a second. Okay, so as an overview, when you have low substrate concentration, the Km is larger than the substrate concentration. And at this point, the more substrate that you add, the higher the velocity. So the velocity depends on the substrate concentration when you have a low substrate concentration. So that's what this line signifies. The slope is almost straight. Okay, and the second situation is when you have the substrate concentration equal to the Km. So that's right here. So at this point, that's half the maximum velocity. So half of the enzyme binding sites are full. And then lastly, if you go all the way to the right-hand side on the x-axis, that's the highest amount of substrate concentration. And here you can see that when you start off with a lot of substrate, you can see on the x-axis that the Km is smaller than the substrate concentration. So yeah, your substrate concentration is way over here on the right, and your Km value is way on the left. So your substrate concentration is higher than your Km value. The Km becomes negligible, and the equation can be simplified to V naught is equal to V max. And basically all of your enzyme is saturated. No matter how much substrate you add, your velocity will not increase. So here we're talking about number 3 now. Which one of the following is true for an enzyme that follows Michaelis-Menten kinetics? Okay, so A, enzyme substrate concentration is larger than free enzyme if the substrate is smaller than the Km. So we went over this situation, it was the first one. When the substrate concentration is smaller than the Km, this means that not even half of the enzyme binding sites are filled with substrates. So the enzyme substrate concentration would be smaller than the enzyme concentration alone. But here it's saying that it would be larger, so that's not the case. It would actually be smaller. Okay, now C. Doubling the concentration will have a large effect on the rate. So we went over this too. When you double the concentration, it has a large effect only at the beginning. So when the substrate concentration is smaller, only right here. When the substrate concentration is smaller than the Km, that you can see through this steep slope, that if you double the concentration there, it would have a large impact. So this is saying that if the substrate concentration is larger than the Km, and that's not true. That is only true if it is smaller than the Km. And then D, half of the active sites of enzyme are occupied by substrate if the substrate concentration is smaller than the Km. So we went over this, and that's not true. Half of the binding sites are occupied when the substrate concentration is equal to the Km. So now the correct answer, the rate is not proportional. The rate is not proportional with substrate concentration if the substrate concentration is larger than the Km. So let's look at our graph. So we found out here that if the substrate concentration was smaller than the Km, then the rate was dependent on substrate concentration. But once you pass the Km value, like let's say you're over here, at that point, the rate is not proportional with substrate concentration because you can add more and more and more substrate, and this has less and less and less of an effect on the velocity because the enzymes are already getting full. The enzyme binding sites are getting full. Okay, for number 4, these are just facts that you should know based on lecture PowerPoints. So make sure you go over all your lectures and really try to learn those. Okay, which of the following are true? So they want true for enzyme regulations. 1. Homotropic allosteric enzymes do not follow, they do not follow hyperbolic Michaelis-Menten. So hyperbolic, they're just talking about this right there. Michaelis-Menten relationship. So this is true. And phosphorylation of glycogen phosphorylase is an example of regulation with a covalent modification. That is true. And then regulation of chymotrypsinogen with trypsin is reversible. So this is not true. So the answer is 1 and 2. And then number 5, the figure below shows the first step of the peptide breakage mechanism by chymotrypsin. Which of the following is false for the mechanism? So right here on the left, I added some definitions for you. A, the hydrophobic pocket. So this is A, the hydrophobic pocket. And this is where the aromatic side chain of the substrate will bind. So that's this guy, this aromatic side chain. Remember that chymotrypsin requires an aromatic chain in order to be able to break this peptide bond. So whenever the substrate enters the active binding site, this aromatic side chain will be placed inside of the hydrophobic pocket. And then B, that's the oxyanion hole. So the oxygen from the carbonyl, so now they're talking about this oxygen right here from this carbonyl, that's actually going to be placed in here during one of the steps when that oxygen acquires a more negative charge. And so what happens is that the reason that it's placed in this oxyanion hole is because this the amide from the glycine and the serine, so these hydrogens here, are going to stabilize that partial negative charge on the oxygen. So that's the hydrophobic pocket and the oxyanion hole. So let's see, number 5 question. Histidine 57 involves in acid-base catalysis. So yes, this is true. Let's see, what were they asking? Oh, they want to know which one is false. So C is false. So I'm just going to put true right here. Okay, so histidine 57 involves acid-base catalysis. Yes, it does. Okay, B, region labeled as A in the figure stabilizes the aromatic side chain of the peptide. Yes, it does. We just went over that. D, aspartic acid 102 and histidine 57 interactions cause an increase in pKa of histidine. So this is true. Histidine's pKa value goes from 7 to 12. So it becomes a strong Lewis base and it steals the proton from serine. And that's what makes serine a strong nucleophile. And then C, glycine 193 acts as a strong nucleophile. So this is false. So that's false. Because we just talked about what glycine does, and it actually helps to stabilize that. So they're talking about this glycine, and that we talked about it. The hydrogen stabilizes that oxygen that's going to have a negative charge, and the nucleophile is serine, not glycine. Okay, so which of the following is true? Sucrose is a reducing disaccharide made up of glucose and fructose. So that's not true. So I'm giving you a lot of vocabulary, so we're going to go over that first. So a reducing sugar must have a hemiacetal group. There we go, hemiacetal group, so that it can be oxidized. And so this is a hemiacetal group right here. So you can see that we have a carbon, and there has to be an OR group and an OH attached to the same carbon. That is a hemiacetal group. So if you have any disaccharides and they have this on the outside of the second disaccharide, then that is a free hemiacetal group, and that will be a reducing sugar. So right here on the right, I gave you some monosaccharides. So you have glucose, fructose, mannose, ribulose, and galactose. Should be able to identify those. And then here we have some common disaccharides like sucrose, maltose, and lactose. And we can see that sucrose is made of glucose and fructose. So right here, glucose and fructose. And they said that this was a reducing sugar, and that's not true. Sucrose is not a reducing sugar. And then B is anomeric carbon of a ketose sugar is carbon number 1. So that's not true. It's actually carbon number 2. And then I went ahead and brought over this ketose example for you. So this is ketose. And the way that you can tell that this is ketose is because the carbonyl is not at the end. If the carbonyl, this carbonyl right here, if that was at the end, then this would be an aldose. But because the carbonyl is on carbon number 2, that's what makes this a ketose. And so its ring is going to be different than if it had been an aldose. So for the ketose, the ring actually looks like this. Carbon number 1 is up here, and then carbon number 2 is in the anomeric location. Okay, so this is still the anomeric carbon, and it's carbon number 2, not number 1. Okay, now some more definitions, and you should probably have these memorized. All these definitions you should have memorized. Stereoisomers have the same molecular formula, but different configuration. That's a stereoisomer. Same formula, different three-dimensional structure. So diastereomer, now we're over here. Diastereomers are stereoisomers that are not mirror images of each other. They can differ at 1 chiral carbon or more. So a diastereomer is when two things have the same molecular formula, but the three-dimensional structure is different. So here they're saying that at 1 chiral carbon they can be different, or at more than 1 chiral carbons they can be different. Now an epimer is a diastereomer that differs at only 1 chiral carbon. An enantiomer are stereoisomers that are mirror images of each other. So I remember enantiomer because this is a lot of terminology. Enantiomer, I remember the Harry Potter mirror of Elisad or whatever. So when I remember that mirror, I just think enantiomers, mirror images of each other. I don't know if that'll help you. Anomer. An anomer are stereoisomers and diastereoisomers that differ in configuration at the anomeric carbon. So for an anomer, it's talking about the anomeric carbon specifically. It has to differ at that carbon. Okay, so now we're back at C. So we see that this wasn't true, this wasn't true. C. D-glucose and D-galactose are enantiomers. So I think I put a figure. So here is D-glucose, and here is D-galactose. And we can see that carbon 1 is the same, carbon 2 is the same. The hydroxyl group is on the right for both of them. Carbon 3 is different. So carbon 3 is different. For galactose, the hydroxyl is on the left, and for glucose, oh wait, it's the same. Okay, so that's the one. It's actually carbon number 4. That is different. The hydroxyl's on the right, but for carbon number 4, the hydroxyl's on the left. And then for carbon number 5, they're both on the right. And so now we can go back to the question. D-glucose and D-galactose are enantiomers, and the definition of an enantiomer was that they're stereoisomers that are mirror images of each other. And we know that these are not mirror images of each other because they're the same except that they differ at 1 carbon. So that's false. And then D, alpha D-glucopyranose and beta D-glucopyranose are anomers. And this answer is yes, because they differ at the anomeric carbon. Okay, so if you need more detail about this, I already have a video on YouTube from one of the lectures that I gave, where I give you all of the rules and definitions for when something is alpha, when something is beta, when something is D versus L. So just look, there's a teaching playlist, so it'll be on there. Okay, number 7. Which of the following is false for polysaccharides? So again, we're going to start off with a bunch of definitions over here on the right. So starch is found in plants. So remember, a polysaccharide is just a bunch of sugar monomers. So there's different types. Here we have starch, we have cellulose, we have chitin, and glycogen. So starch stores energy in plants. Starch stores energy in plants. We have 2 types of starch. We have amylose and we have amylopectin. So amylose is a straight chain. There's no branches. And the linkages are alpha 1,4. Now amylopectin is a straight chain with branches. And the branches are alpha 1,6. Okay. So then we have cellulose. Cellulose is also found in plants, but it's for structural support. So we had starch in plants for storing energy, and then we have cellulose also in plants for structural support. So these are beta 1,4 linkages. They're found in the walls. They're tough, fibrous, water insoluble. They're linear. They're unbranched. So cellulose is unbranched. They're homopolysaccharides. It is different from amylose in the linkage. It is beta instead of alpha. So if we go look at our amylose, it was basically the same, but it's alpha 1,4 linkages. And for cellulose, it's beta 1,4 linkages. So then we have chitin, and this is found in insects for structural functions. It's a linear homopolysaccharide made from N-acetylglucosamine residues that have a beta 1,4 linkage. And then we have glycogen. Glycogen is found in animals, and it is highly branched. And it has both types of linkages, the alpha 1,4 and the alpha 1,6. Okay, so knowing all of those facts, we can go ahead and answer a question now. Which of the following is false? So they want false. So we have amylose has long unbranched chains of glucose. So let's look at amylose. Unbranched, yes, that's true. Amylose is straight chain. There's no branches, so yeah, that's true. So that can't be the right answer because they want false. So then C. Both glycogen and amylopectin form branches via the alpha 1,6 linkages. So let's see. Glycogen, that's down here, and it says the branches are alpha 1,6. So for this one, the branches are alpha 1,6 for glycogen. And what about amylopectin? Amylopectin is up here, and it says that the branches are alpha 1,6. That's true. So both glycogen and amylopectin form branches at alpha 1,6 linkages. That is true. So that's the wrong answer because they're looking for faults. D. Chitin is a homopolysaccharide chain connected by beta 1,4 linkages. Let's see. Chitin, homopolysaccharide chain, yes. And then beta 1,4 linkages, yes. So that's true. So that can't be the right answer. And then B, cellulose is highly branched polysaccharide. That makes it a strong material. So we go to cellulose, and it's linear and unbranched. So cellulose was unbranched, and they're telling us that it is highly branched. So that is wrong, which means that it is false, and that's a right answer. Enzymes catalyze reactions by decreasing the activation energy to form the transition state. This is just a fact that you have to know. So I can't really explain it too much. You just, I already uploaded a video on this topic as well, and I go into it in a lot of detail, but you basically, that's the one way that enzymes function. They decrease the activation energy to form the transition state. Number 9, which of the following is true for hexokinase enzyme? So Professor Dudagachi covered about 3 enzymes in lecture, so hexokinase was one of them. So they're looking for true. So let's go to A. Its substrate is glucose 6-phosphate. So this is false. For hexokinase enzyme, the substrate is glucose and the product is glucose 6-phosphate. So students have trouble with this, but let's go ahead and draw it a little bit. So glucose is just like the circular form, and then glucose 6-phosphate, all it is, let's say that this is carbon number 6 right here. The enzyme, all it does is add a little phosphate. So that's now glucose 6-phosphate, and that's what hexokinase does. So that is not true because they had it backwards. B, it uses free ATP to catalyze the reaction. So that's false because ATP is in a complex with magnesium when it is used. And I went ahead and copied the notes from your lecture pertaining to this topic. Hexokinase is a glycolytic enzyme. Hexokinase is a bisubstrate enzyme that catalyzes the reversible reaction, so it's reversible, from beta-D-glucose to glucose 6-phosphate, while ATP binds to the enzyme as a complex with magnesium. So remember, whenever you have a complex, I had some students not really understand that term. Complex just means that they're together, so ATP and magnesium are together. And the question was saying that ATP was free, and that's not true. It's in a complex. Okay, so now D. The enzyme cannot discriminate between glucose and water. So the enzyme can discriminate, so that's not true. The enzyme can discriminate. So I went ahead and copied some more notes from lecture for you. They're down here. In the hexokinase reaction, the gamma phosphoryl of ATP is transferred to the hydroxyl at carbon 6 of glucose. This hydroxyl is similar in chemical reactivity to water. The enzyme can discriminate between glucose and water because of a conformational change in the enzyme when the correct substrate binds. This is a good example of induced fit. Water binding cannot cause induced fit. Okay, and we can go to C. Induced fit conformational change takes place when the correct substrate binds. So we know that this is true for hexokinase enzyme. Okay, number 10. Which of the amino acids below is less likely to involve in acid-base catalysis? Okay, so you need to know the 3 forms of catalysis. There is a video on my playlist. I didn't record it, but I saw it and I loved it. It's like 5 minutes long, and they do a beautiful job of explaining the 3 types of catalysis. So watch that if you need help with that. Also, you have to know your amino acids, which ones are ionizable, because they're the ones that can participate in acid-base catalysis, because they're the ones that have pKr values and can donate their protons. So by now you should know like all of those by memory. And so if you do, then you know that histidine, aspartate, and arginine they all can participate in acid-base catalysis. Alanine is a nonpolar amino acid, so it can't participate. Yeah, also polar, so like polar, and then the ones with pKr values. Just know your amino acids and their categories. Okay, and then you should be able to recognize like the most common sugars. So that's why just practice on a whiteboard. Okay, so that was the end of Professor Bedcham's practice exam, and now we're starting Maria Zogby's exam. So here for number 7, select a sentence that correctly describes the following molecule. So I just recorded a whole lecture on this. I think it was like 45 minutes long. So it's on the channel already if you are struggling with this. So glycerol is one of those molecules that you just have to know how to draw. You have to have it memorized. So there's carbon 1, 2, and 3, and each one is attached to a hydroxyl. And then you just fill in the hydrogens. So this is glycerol. And then we see here on the backbone, we have glycerol, and it's bound to 3 fatty acids or 3 acyl chains. And each one of these has a different number of carbons. So I went ahead and numbered the carbons already, 1 through 16 for the first one, 1 through 18 for the second, and 1 through 18 for the third. Now this triangle with a number, remember that tells you the location of the double bonds. So this triangle with number 9, that just means that at carbon 9 we had a double bond, which we did. And here this triangle with 9, 12, and 15 means that we had a double bond at 9, 12, and 15. Okay, so select the sentence that correctly describes the following molecule. A, contains sphingosine as the backbone. So this is sphingosine right here. It's another molecule that you also have to be able to identify and draw, or at least identify. So you can see that it has 3 carbons, carbon 1, carbon 2, and carbon 3. To carbon 3, you just have an alcohol attached. Carbon 2 has an important group, this NH2. So this is important because it will form an amide linkage when something attaches. And then to carbon 1, we have an alcohol on one side, and then this really long carbohydrate, carbon and hydrogen chain. And then these guys can switch places, so don't be confused if you see it on the right-hand side all of a sudden. Okay. So, does this molecule contain sphingosine as a backbone? The answer is no. And the way that you can tell is that there's no nitrogen at carbon number 2. Okay. So you know that this is glycerol. Okay, so A is not true. B, contains an amide linkage. Nope, there's no nitrogen there, so there's no amide linkage. And then C is an amphipathic molecule. So this is a triacyl glycerol. So this is let me write it down. So glycerol is the backbone, and then it's got 3 fatty acid chains. So the appropriate name for this is a triacyl glycerol. And you can see here on the left it's just carbons, and then on the right it's just carbons and hydrogens. There's no polar groups here, so this is not an amphipathic molecule. It's actually a hydrophobic molecule. So a triacyl glycerol is a hydrophobic molecule. There are no polar groups here, so it is not amphipathic. D has a 16, 3, delta 9, 12, 15 chain at carbon 2. So let's look at carbon number 2. That's this one right here. And it says, remember this 16? That is the number of carbons. But we see here that our second carbon has 18 carbons, not 16. And then it has 3 double bonds at 9, 12, and 15. So that's not true. This second carbon only had one double bond at carbon number 9. So that's what makes this wrong. And then E, none of the above. So that is the correct answer. So you really have to know your stuff here. Okay, number 8. Which of the following molecules will form micelles in aqueous solutions? So here's some notes from your lecture. We have individual units are wedge-shaped. Cross-section of the head is greater than that of the side chain. So you can see that polar head group, the cross-section is larger. Okay. Which of the following molecules will form micelles? Okay, so A, triacylglycerols. No, these are hydrophobic. We just talked about these. So you can see that this is a triacylglycerol. First of all, there's 3 tails, not 1. And secondly, this entire molecule is hydrophobic. So that's not going to work, because in order to form a micelle, you got to have a polar end, which is like the head right there, and then a nonpolar tail. That's the hydrocarbon chain. And if you're struggling with polarity, definitely go back and watch video number 1. I have a 10-minute slide that explains the concept of electronegativity, and then after that you'll hopefully really understand polarity for the rest of the semester. Okay, so sphingolipids, can they form micelles? The polar head is not greater than the side chain. So did I bring in a sphingolipid? I don't know if I did. But basically a sphingolipid will have like a sphingosine as the backbone. So no, that's like way too big. It won't form a micelle. And then we have sterols. So sterols are a ring structure. So no. And then E, glycerophospholipids. So this is an amphipathic molecule because of the phosphate. And it has a glycerol backbone and phosphate. So that would be amphipathic, because this part is nonpolar and then this part is polar. So that makes it amphipathic. But it has too many tails, because here the rule is that you can only have 1 tail if you're going to form a micelle. So that's why E is not the right answer. The right answer is fatty acids, D. So we can see like this right here is 1 fatty acid, and so that can form a micelle. And this would be like the polar head group, and then this would be the hydrophobic nonpolar tail. Okay. And then here is like an important thing that I thought you guys should know. We have membrane lipids, which is in this red box. And then we have storage lipids. So for storage lipids, we have a triacylglycerols. And this is what we just went over, the glycerol molecule with the 3 fatty acid chains. So those are triacylglycerol, and they're storage lipids. And remember that they're hydrophobic. Okay. And then membrane lipids, we have 3 different kinds. You can have phospholipids, which means that they have phosphate. You can have glycolipids, which means that they have a sugar, or you can have archaeolipids. Now, for phospholipids, you can have 2 different types. You can have glycerophospholipids, where glycerol is in the backbone, or you can have sphingolipids, and that's when sphingosine is in the backbone. And then, the glycerophospholipid has 1 fatty acid tail, another fatty acid tail, the phosphate, and a polar head group. And then the sphingosine will only have 1 fatty acid, and then the phosphate, and a polar head group. And then for membrane lipids, glycolipids, the sugar lipids, you can have sphingolipids and galactolipids, and these are the sulfolipids. So for sphingolipids, same thing. Sphingosine is the backbone. You have 1 fatty acid, but instead of the phosphate and the choline, now you have a mono or an oligosaccharide. So you have sugars. And then for a galactolipid, you have glycerol as the backbone, 2 fatty acids, and then sugars, and then a sulfate group. And then you have the archaeolipids. So you should be able to just know like the backbones for those categories, just in case. And then I had a student ask me about general types of membrane lipids, so I made this note right here. There's glycerophospholipids, galactolipids, and sulfolipids, archaeals, sphingos, and sterols. Okay, so number 9, which of the following membranes would be the less fluid? So they want the more rigid. Okay, so you have to know the rules. So there's, what are the factors that affect membrane fluidity? So we have temperature. So we have temperature is a factor. Let me use blue. So temperature affects membrane fluidity. Length of the tail affects membrane fluidity, and saturation of the fatty acid, whether it's saturated or unsaturated, that affects membrane fluidity. So here they want less fluid, so decrease fluidity decreases, and low fluidity. So low temperature, saturated, and longer tails, that will decrease fluidity. And remember that cholesterol plays a dual role. So when the temperature is cold, when you have low temperature, cholesterol helps to increase fluidity. And when you have high temperature, when it's too hot, cholesterol helps to reduce fluidity. That's just a fact that you have to know. But to answer our question, they want the more rigid, the less fluid, so that we should have lower temperature, saturated, and longer tails. So here we have 18, 18, 16, and 16, but we know that we want the longer tails in order to be less fluid. So we immediately cancel out the lower, the 16. And then we know that we want saturated, because saturated decreases fluidity. So here we have saturated and we have unsaturated. So we definitely can get rid of the unsaturated. And all of the above are equivalent in fluidity. We can delete that because we know that's not true. And so by the process of elimination, B is our answer. That's the less fluid. Number 10, an integral membrane protein can be extracted with a solution containing detergent. Remember that detergents are amphipathic, so they have a polar head group and then a non-polar group, and this can disrupt the membrane so that the integral proteins can be released. So scientists are still struggling with removing membrane proteins because they're so protected in the bilayer, and in that hydrophobic bilayer, the proteins are so protective. So yeah, you have to use a detergent like SDS, which was sodium dodecyl sulfate, is a popular one. Okay, so number 11, when an uncharged molecule, moves from a concentration of 10 to the minus 4 to 10 to the minus 2, is the process spontaneous? Is it at equilibrium? Or does it require an energy? So let's look at this. So 10 to the negative 4 molarity, I just like to write it in decimal points because it's easier for me. So it's 0.0001, and then 10 to the minus 2 is just 0.01. So this is lower concentration and this is higher concentration. So you have a molecule going from low to high concentration. So when you go from low to high, you are going against the concentration gradient, and this means that it is not spontaneous and it will require energy. So this is just knowing like your vocabulary. So input of energy is required. This is not at equilibrium. This is not spontaneous. It does not depend on membrane potential. And one of those is correct. Number 12, select the option that best describes the transport proteins involved in the process shown below. So up here we have different types of proteins. So we have uniport, so that means only one substrate moves along. And then we have co-transport, when two substrates move at the same time. So we have symport, that means both substrates go in the same direction. And then we have antiport, and that's when one substrate moves in one direction and the other substrate moves in the opposite direction. Okay, so here we have the sodium potassium ATPase they're saying that it's a channel that allows the electroneutral symport of these ions. So this isn't true. The sodium potassium ATPase is a pump, and it allows charged antiport of these ions. So you can see it here, sodium potassium ATPase pump. You can see that potassium is charged and it's going to the left, and you can see that, what else? Sodium is also charged and it's going to the right. So that's why it's antiport, and it allows charged, and it's a pump. Okay, so B, the sodium glucose transporter is responsible for maintaining the negative membrane potential of the epithelial cells. So this is a secondary active transport, and this is the sodium glucose here. So sodium is positively charged and it's going into the cell, so it can't be responsible for maintaining negative membrane potential. And then C, the glucose flow from the interstitial lumen, so that's right here, glucose flow from the interstitial lumen to the blood, so from the lumen to the blood. So the glucose flow from the lumen to the blood requires secondary active transport and facilitated diffusion. So yeah, that's true. Okay, number 13, calculate the total free energy change for transporting 1 mole of sodium out of the cell when the membrane potential is negative 70 millivolts. So remember, the first thing I do is convert from millivolts to volts. So that would just be 0.07 volts. And the concentration of sodium is 10 millimolar inside and 140 millimolar outside. So the first thing I like to do is draw. So I'll draw it right here. So we have the cell. This is outside. This is inside. And so they said that they're transporting sodium out of the cell. And they're saying sodium inside is 10 millimolar, and outside it is 140 millimolar. So it's going from low to high. So anything that moves from low to high concentration is against the concentration gradient. Okay. And they're talking about sodium, and sodium has a positive charge, which means that we have to use this equation right here. So delta GT, that's the free energy change for transport. So if you are transporting a molecule that does not have a charge, then you would only use this green portion, the chemical gradient portion of the equation. That's R, that's the universal gas constant, times T, that's temperature in Kelvin, times the ln of the concentration 2 over concentration 1. Concentration 2 is the concentration, that's the final destination. So in our case, sodium is being transferred out of the cell. So this would be concentration 2, because that's the final destination for sodium. And this would be concentration 1, because that's the initial destination. Okay, So remember, if the molecule is not charged, we would only use this chemical gradient portion of the equation. But because sodium has a positive charge, we also have to incorporate the electrical gradient portion of the equation. So you just put a plus sign and you put Z, F, delta, and then I don't know what that little guy is called. But Z is just the charge. So in this case, sodium has a +1 charge. So your Z value will just be 1. F is Faraday's constant, and it's right here. 96,500 joules per volt times mole. And then delta, that guy, that's just the membrane potential, transmembrane electrical potential in volts. So we had already converted to volts. Okay, so now we're going to go ahead and do the math here with the information that we were given. So practice this math a couple of times. It's actually really easy, but you want to make sure you know how to do it. So delta G is equal to the universal gas constant, so that's just 8.315 joules per Kelvin times mole times 310 Kelvin. They gave us the temperature in Celsius, so you have to be able to convert to Kelvin. Ln of 140 over 10, so this was concentration 2 over concentration 1, and then plus, this was our Z, the charge for sodium was +1. This is F, that's the Faraday constant, 96,500 joules per volt mole. And then this was the transmembrane electrical potential, this guy. So that was 0.07 volts. And then when you multiply, you end up with 6803 joules per mole for the chemical gradient portion of the equation, and then 6755 joules per mole for the electrical gradient portion of the equation. So once you have that, you just add them together, and that gives you 13,557 joules per mole. But delta GT is always given as kilojoules per mole. So you just move the decimal point over 3 times, and so that will give you 13.55. So you just round up to 14 kilojoules per mole, and that's how you do the math. It seems a lot, but once you know the terminology, it's actually not bad. Okay. For number 14, the figure below shows the cycle for the sarcoplasmic endoplasmic reticulum calcium pump. If the calcium concentration in the lumen of sarcoplasmic endoplasmic reticulum is 1 millimolar and 100 nanomolar in the cytosol, where the A, P, and N domains are, what sentence best describes this pump? So I just went ahead and converted these because they were from millimolar and then nanomolar, and I don't like that. So this is what they are. In the lumen, it's 1 times 10 to the negative 3 molar. And then in the cytosol, it's 1 times 10 to the negative 4 molar. Okay. And then, here we go. Calcium is transported from the lumen to the cytosol. So that's not true. The sarcoplasmic reticulum is in the cytosol, and calcium is transferred to the lumen. So they had it backwards. And then B, the Circa pump belongs to the ABC transporters family, so it doesn't. It belongs to the P-type ATPases. C, phosphorylation of the Circa pump activates calcium transport. It doesn't inhibit it, so that's why that's wrong. D, the E1 conformation, so that's up here. The E1 conformation has high affinity for calcium binding sites, whereas the E2P conformation has low affinity. So this makes sense because at high affinity, like the high affinity for calcium means that it holds on to calcium. So in the E1 conformation, nothing has been phosphorylated, so it makes sense that it would want to hold on to the calcium. So it would have a high affinity for it. But in the E2, this is after phosphorylation. So it makes sense at this point that you would have conformational changes in order to release the calcium. So you would want to have a lower affinity to release the calcium, because that was the whole point. And then, so that's why that's correct. Now E, the E1 conformation has low affinity. So here they give it to us backwards. So remember, low affinity would mean that it would release it. So this would be saying that you haven't even phosphorylated yet, and you're already going to go ahead and release the calcium. But that's not how it works. It has to bind calcium tightly, and it'll only release after phosphorylation. Okay. Number 15, the extracellular sodium concentration is 145. So again, I started drawing. For these types of problems, it's really important to start drawing. So here on the right, you can see that we have our cell. This is outside and this is inside. And they're saying the extracellular sodium concentration is 145. So yeah, 145. And then inside, it is 5. And the membrane potential is minus 60 millivolts. So that would be 0.060 volts. So they're saying if a sodium channel, so now we're not talking about a pump, we're talking about a channel. If it opens, in which direction will the sodium move based on both its electrical and chemical gradient? So here I went ahead and copied from the notes what a channel and what a transporter are. So for the channel, we have the rate is near diffusion limits. It is super fast for a channel. The saturation for a channel, it does not saturate. And the gating mechanism for a channel is that one gate is opened by a biological signal. So ions flow down their electrochemical gradient. So that means that they go from high concentration to low concentration. And that's what we have here. We have a channel. And so we have high concentration outside and low concentration inside. And it happens really fast near the rate of diffusion, and things can't be saturated. So the correct answer is C. Sodium will spontaneously enter the cell. It moves in favor of its electrochemical gradient from high to low concentration. Now, let's talk about why the other answers are not correct. A, the chemical gradient tends to move sodium into D cell, but the electrical component opposes such movement and there is net flow of sodium out of the cell. So that's not happening. It's moving from high to low concentration. And then C, B. Sodium tends to move inside the cell, but potassium tends to compete for the selectivity filter in the pore of the channel, inhibiting the net transport of sodium. So they're talking about something completely different here, not a channel. So that's why that is false. Yeah, they're talking about the sodium-potassium pump. Okay, so now D. Sodium won't be able to move unless an energy source is provided. So no, when you have a channel, things just naturally move from high to low concentration. You don't need energy. E, none of the answers is correct. So no, C is correct. Okay. And that's it. That's the study guide for chapter 2. So I'm going to have office hours on Sunday for 2 hours. You can find them on Zoom. So if you have any last minute questions, you can come over and ask. I think I'm going to have them Sunday morning from 9 to 11 a.m. Good luck on your test.